Today we're going to discuss the oxidation of glucose, which means the breakdown of glucose. We're going to systematically break down those carbon-carbon, carbon-hydrogen bonds to release energy in the form of electrons and hopefully harness as much of that energy as we can in the form of ATP. So we're going to systematically go through, break down glucose to form pyruvate, our 2,3 carbon molecules. We will then convert that pyruvate into acetyl-CoA, and then I will discuss what happens once acetyl-CoA enters into the citric acid cycle, also known as the Krebs cycle. So when we're talking about the breakdown of glucose, it's really so that we can obtain energy from organic molecules. And we could be talking about other organic molecules such as proteins or fats. But we use glucose as a way to really study these pathways and how we're converting the breakdown of those molecules into ATP. As we've already discussed, we're going to be able to utilize the energy stored in ATP to drive numerous endergonic reactions from within the cell. Most of what we'll be talking about as it pertains to humans will be aerobic cell respiration, in which we're utilizing oxygen and producing CO2 as a byproduct. Oxygen is going to act as the final acceptor of the electrons that are released from the glucose. At the end of this chapter, we'll also talk about anaerobic cell respiration, which is utilizing alternative terminal electron acceptors to ultimately take up those electrons once glucose is broken down, but it will not be oxygen that's going to accept those electrons. And that's in contrast to fermentation, which is what happens in the absence of oxygen, but when you cannot use an alternative terminal electron acceptor. So we'll differentiate all that towards the end of this chapter. So we talk about the breakdown of glucose as occurring in 4 major metabolic pathways. The first is glycolysis, and the name tells you what it's going to undergo, which is the lysis of glucose. We're going to literally break the glucose molecule in half. We'll then talk about the breakdown of pyruvate, and sometimes this is referred to as the oxidation of pyruvate into acetyl-CoA. The third step is known as the citric acid cycle. The completion of that will result in a complete loss of all of our carbons. We're going to, again, start with glucose, which is C6H12O6, and by the end of the citric acid cycle, all of our carbons will have been eliminated in the form of CO2, which you will just exhale. However, by that point, we have been able to generate a lot of high energy electron carriers that are going to take those electrons to the electron transport chain, where oxidative phosphorylation will take place, and that's where we will ultimately make the bulk of our ATP. So the first step, glycolysis, that's taking place in the cytosol. However, steps 2, 3, and 4 are all taking place in the mitochondria. Now, as we progress through these next 4 steps, you need to be able to keep an eye on what's happening in the big picture for each of these main steps, in terms of how much ATP is being generated, and not just how much ATP is being generated, but also how is it being generated. We'll differentiate between substrate level phosphorylation and oxidative phosphorylation. You also need to be able to determine how much NADH and FADH2 are being produced in each step. These, again, are known as our high energy electron carriers. And then lastly, I want you to keep track of your carbons. So we're starting with C6H12O6 and what's happening to those carbons in each of the 4 steps that we will go through systematically. So you've been provided with a table, and as we go through this content, you should be filling this out and basically be able to regenerate this table on your own just from memory. All right, so let's begin with glycolysis. And again, big picture, all we're doing here is taking a 6-carbon molecule, glucose, and the end result after 10 enzymatic steps will be the production of 2 pyruvate molecules, 2 3-carbon molecules. So we're starting with 6 carbons and we're actually going to end with 6 carbons so we don't lose any carbons in the process of glycolysis. So before we jump in, I just want you to keep in mind this big question. So we know that cells comprise our bodies and they are highly ordered. Yet, when we talked about some of the laws of thermodynamics, you know that the universe is always moving towards a state of greater disorder. So how is it that our bodies are able to maintain this order in spite of the second law of thermodynamics? So take a second and I want you to go ahead and answer this question. Now, even though the second law of thermodynamics would say that we are moving towards a greater state of disorder, the first law of thermodynamics argues that energy can neither be created nor destroyed, but it can be transferred. So as we are breaking down our different macromolecules, in this case, as we're breaking down glucose, all that energy does not have to be completely lost in the form of, say, heat, but if we can transfer some of that chemical energy from glucose, from those carbon-carbon, carbon-hydrogen bonds, and transfer it to another chemical, namely ATP, then we can efficiently store some of the energy that would have been lost from the breakdown of glucose. Here we are showing what the free energy is for the reaction as we go through the complete oxidation of glucose, and the delta free energy is minus 686 kcals per mole. Now, to form a single ATP molecule, again going from ADP and adding a phosphate group to that to make ATP, does take 7.3 kcals per mole. And as we're breaking glucose down, we are able to generate somewhere between 34 and 38 ATP. Now, this process is going to take some energy, so we never really make 38 ATP. But let's assume, for the sake of argument, that we can make 34 ATP. How much of the potential energy that is in glucose is harnessed in the formation of ATP? So I want you to do the math. Go ahead and get out your calculator and figure out how many kcals per mole are we able to generate if assuming that we're making 34 ATP. So if you did your math right, we are generating 248.2 kcals per mole. And so it's about almost 40% efficient, which is actually not bad. So even though a decent amount of the energy as we're breaking down glucose is lost to the universe, we are able to harness some of that energy that we can then use to drive other endergonic reactions in the body. Now, before we go through the actual steps of glycolysis, I want to review a few key terms and then we'll get down to the specifics. The first is how we define cellular respiration. This is the catabolism, or the breaking down of high energy molecules, namely for us from glucose, that's going to result in the production of energy, namely ATP. And the important thing to point out here is that you should be associating the term respiration with the utilization of the electron transport chain. So obviously this can take place in the presence of oxygen, where oxygen is going to act at the end of the electron transport chain as the acceptor of the electrons that originally came from glucose. And so that would be aerobic cell respiration. But there's also processes known as anaerobic cell respiration, which would be true for certain types of bacteria. So again, they're not going to use oxygen as the terminal electron acceptor, but they can use other things that are able to take those electrons, such as nitrates, sulfates, carbonates, etc. And we'll look at that in the next lecture. And anaerobic respiration is in contrast to fermentation. So fermentation does not use the electron transport chain. So this, like anaerobic respiration, is not taking place in the presence of oxygen. However, we don't use the ETC. Instead, we are going to shuttle pyruvate into an alternative pathway. And we'll talk about that process as well. All right, so let's talk about cellular respiration in the presence of oxygen. So the very first step is going to be glycolysis. And what we're going to be doing here is breaking down glucose to make ATP, and this can occur whether we have oxygen present or not. And so we have 10 steps in glycolysis, and we have them divided into 3 main phases. So the first step in breaking down glucose is actually what we call energy investment. And so we're actually going to be putting more energy into the glucose molecule. Then we will cleave that molecule. We're going to break it in half, and then we're going to harness energy off of the glucose molecule. All right, so that first energy investment stage is really the first 3 steps of this 10-step process. And we're going to break ATP down and take a phosphate group and attach it onto the glucose molecule. Again, in that next step is the cleavage step. These are steps 4 and 5, where we're going to take our 6-carbon molecule and break it into 2 3-carbon molecules. And then the last steps in glycolysis is where we liberate the energy, and we're going to be at that step making 2 NADHs. And again, I defined NADH as a high energy electron carrier, and then we're going to be making 4 ATP. So we initially, during our energy investment step, it cost us 2 ATP. We're making 4 ATP, so we get a net of 2 ATP during glycolysis and 2 NADHs, 2 high energy electron carriers. Here's that first phase, the energy investment phase, and what you can see is we've got glucose, and we're going to actually break down 2 ATPs to make ADP. And where did those phosphates go? They actually are attached onto glucose. So now we end up with something called fructose 1,6, because they're coming off of the 1 carbon and the 6 carbon, bis, which just means 2 phosphates. So we've now added 2 phosphate groups from 2 ATP molecules. So at this point, we have not generated ATP. This has cost us 2 ATP. So this so far is energetically unfavorable. We are then going to go through the cleavage steps. So we had a 6-carbon molecule coming into this. We're going to now generate 2 3-carbon molecules, and they each have 1 phosphate group still attached to it. So we're going to start now at this point to be harnessing and liberating the energy off of this molecule. And that is seen in these final steps here. We're going to start to break off those phosphate groups and actually make some ATP, as well as generate some NADH, our high energy electron carriers. So let's look at some of the specifics of this reaction. Again, we're starting with glucose, which is a 6-carbon molecule, and we will end with 2 pyruvates, which are 2 3-carbon molecules. So again, we've not lost any carbons at this point. We do generate 2 ATP when 2 ADPs and 2 inorganic phosphates come together. So adenosine diphosphate along with the phosphates can become adenosine triphosphate. And then we are generating 2 NADHs from NAD+. Okay, so I want to actually show you some of the specific parts of this reaction. So big picture, again, we want you to basically understand that 2 pyruvates are made. We're making 2 ATP and 2 NADHs. But I want you to see that you could actually look at the specifics of these reactions and understand this process. So all I've done here is blown up this one part of the reaction, looking at this particular 6th step in the process. So what I want you to do is take a minute and look at the glyceraldehyde 3-phosphate, which will become 1,3-bisphosphoglycerate. And I want you to identify the two things that happened to this molecule. What's different between this molecule and this molecule? So hopefully you were able to identify the fact that a phosphate group got added on, which you can see here, an inorganic phosphate group was attached. And then we also lost this hydrogen. And so one thing to recognize is, where did that hydrogen go? Well, that hydrogen, along with these electrons, got transferred to NAD+. And if you recall, we are referring to NAD+ as a high energy electron carrier. It's literally carrying electrons, and not just any electrons, high energy electrons that are going to be able to do work for us later on. So even though we've broken down this part of what originally was our glucose molecule, and it's slowly getting oxidized, again, oxidation is losing electrons. Where did it lose that electron? It lost it here off of the glyceraldehyde 3-phosphate. And where did that electron go? Well, it actually was obtained by NAD+ to become NADH. So now NADH has been reduced. Reduction is gaining, well, NAD+ got reduced to become NADH. Reduction is gaining electrons, and it now has more energy, okay? And that energy is going to be able to do work for us later on. And where is it going to do work? It's going to do work for us at the electron transport chain, the fourth step in this process. And so here I've included what's actually happening to NAD+, which is it's gaining that hydrogen atom, and it's also gaining another electron. So NAD+ becomes NADH, and it's actually gaining 2 electrons in that process. And sometimes students really struggle with this, so if this makes it easier to literally think of it as a transporter, the NAD+ is acting as a taxi and literally carrying the electron to another part of the cell. So it's NAD+, it's now carrying that electron. Where is it carrying it to? It's going to carry it to the electron transport chain, step 4 in our overall breakdown of glucose. All right, so let's take a second here and just make sure that you are tracking with all this information. So the question is, NAD+ is an important carrier of high energy electrons in the cell. Which is the oxidized form, NAD+ or NADH? Go ahead and answer this question on the Ed Puzzle. Hopefully you selected A. A is the oxidized form of NAD+ or NADH. Because again, oxidation is losing electrons, and which form is lacking the extra electron? That's the NAD+. NADH was the form that had gained the electron from the breakdown of glucose. And an easy way to think about this, for some students this is easier, is to just track your hydrogens. If you think about a hydrogen atom, it's going to have 1 electron. So if you can keep track of your hydrogen atom, then that's another way to keep track of who has the electron. Lastly, before we move on, I want you to look at what is taking place in this last, the 10th enzymatic step as we've broken down glucose, and just prior to forming pyruvate. I want you to look at what this enzyme, pyruvate kinase, is able to do. So pyruvate kinase is shown here in blue. This would be called an apoenzyme, and it's now interacting with an inorganic cofactor known as magnesium, and that makes our holoenzyme. And this is where our substrate, which is phosphoenolpyruvate, is able to bind. So phosphoenolpyruvate, I want you to look at its structure and look what's different between phosphoenolpyruvate and pyruvic acid. So go ahead and answer that question on the Ed Puzzle. Hopefully you were able to identify that pyruvate lost the phosphate that was initially on this phosphoenolpyruvate substrate. And where did the phosphate go? It was transferred to ADP, adenosine diphosphate, to become adenosine triphosphate. So as we've broken this down, that is going to liberate some energy, free up some energy. Where did that energy go? Did it get lost to the universe? No, we tried to harness some of that energy and form ATP, which takes energy, but not as much as was liberated by breaking off this phosphate from the PEP. And this is known as substrate level phosphorylation. The substrate itself is causing the phosphorylation of ADP to form ATP. It's literally taking the phosphate from the substrate and becoming ATP. Now, this is going to be distinct from what we see later on in at the electron transport chain, which there we're going to be making ATP by a process known as oxidative phosphorylation. So again, as we're generating ATP, as we're attaching on this third phosphate group, we can do that either by substrate level phosphorylation, oxidative phosphorylation, or photophosphorylation, which is something that we will talk about more when we talk about photosynthesis. All right, so at this point, you should be able to fill out the first portion of your table. So again, glycolysis, where is this taking place? For eukaryotes, it's taking place in the cytoplasm, and prokaryotes, they have no membrane-bound organelles, so it's taking place in the cytoplasm for them as well. What was our initial starting material? We had a 6-carbon molecule, glucose. By the end of those 10 enzymatic steps, we had 2 pyruvates, 2 3-carbon molecules. We were able to generate a net of 2 ATP and 2 NADHs, 2 high-energy electron carriers. So now the pyruvate is going to become our starting material in step 2. So let's go ahead and now that you've filled out the table, let's answer a series of questions on the Ed puzzle. Go ahead and answer this first question. If the first stage of glucose oxidation by cell respiration, glycolysis breaks down glucose into what? Hopefully you selected A. It's going to break it down into 2 pyruvate molecules. All right, so next question. Glycolysis is a 10-step metabolic pathway. How many different enzymes are required to convert 1 molecule of glucose into 2 molecules of pyruvate? All right, the correct answer was D, 10. Again, each of those different enzymes is going to be able to recognize a different distinct substrate in that biological pathway. So recognizing that, then our question becomes, how many molecules of pyruvate will be made from 2 molecules of glucose? The correct answer is C, 4. So if 1 molecule of glucose makes 2 pyruvates, then 2 molecules of glucose will make 4 pyruvates. Next question, how many net molecules of ATP will be made from 10 molecules of glucose during glycolysis? The correct answer is E, 20. So if 1 molecule of glucose makes 2 ATP, then 10 molecules of glucose will make 20 ATP. So now that the pyruvate has been made, what we need to do is translocate that pyruvate into the mitochondria itself, which is where all the remaining steps are going to take place as we continue to break down that what had been that original glucose molecule. So we need to get our pyruvate into the inner mitochondrial matrix, which is where it's going to get converted into acetyl-CoA, which is the starting material for the Krebs cycle. So as we break down pyruvate, or sometimes you'll hear it referred to the oxidation of pyruvate, which is really the second step of cellular respiration, what we're doing is, again, transporting the pyruvate into that inner mitochondrial matrix, and it's going to be broken down by a large enzymatic complex known as pyruvate dehydrogenase. And again, anytime you hear the word ACE, you should be thinking that this is going to be an enzyme. And what we're going to liberate in this process is a CO2 molecule, and the remaining byproduct is going to be something known as acetyl-CoA, and that's going to be attached to something called coenzyme A temporarily, and then that will enter into the Krebs cycle. The other thing that is generated in this process is one NADH molecule for each pyruvate. But recall that we have 2 pyruvates entering into this, so we have a net production of 2 NADHs. So let me show you what this whole process looks like. Here we've got pyruvate that was out in the cytosol that needs to get translocated into the inner mitochondrial matrix. And so we're going to do that using a channel protein and a proton pyruvate symporter. Symporter implies that 2 things are getting transported in the same direction at once. So we're going to increase the H+ concentration as we do that here in the inner mitochondrial matrix. So now we've got our pyruvate in the mitochondria itself, and coenzyme A and as well as this sulfhydryl group is going to get attached on to the pyruvate. So you can see that is happening here. We've taken, this was again our initial molecule, and now we've added on the sulfhydryl group and the coenzyme A. But what have we lost? You'll notice that we've lost a CO2 molecule. So this is where we go from a 3-carbon molecule down to a 2-carbon molecule because we've lost one of our CO2s. Now recall again, we had 2 pyruvates. So this whole process is happening 2 times. So we will be generating 2 acetyl-CoAs, 2 CO2 molecules were generated, and 2 NADHs. And where do those CO2 molecules go? Well, as you exhale, you are releasing the CO2 molecules, which are a byproduct of the metabolism of glucose. All right, so take a minute and answer the following question. As pyruvate enters the mitochondrial matrix, it encounters the pyruvate dehydrogenase multi-enzyme complex. Which statement below is false concerning the activity of this complex? A, NAD+ is reduced to NADH. B, CO2 is released. C, coenzyme A is attached to a 2-carbon acetyl group. D, 1 ATP is produced by substrate level phosphorylation per pyruvate. Or E, all of the above are true. So take a minute and go ahead and answer that on the Edpuzzle. The correct answer is D, because D is wrong. We do not make any ATP as we form acetyl-CoA. However, we do make 2 NADHs and we are producing some CO2 in the process. All right, so far what we've done is we've broken down glucose, we've made 2 pyruvates. Those pyruvates have made their way into the inner mitochondrial matrix and have now been converted into acetyl-CoA. We had 2 3-carbon molecules and now we have 2 2-carbon molecules. Where did that carbon go? It was released as CO2 and exhaled, and we did that 2 times. So we've lost 2 CO2s at this point. In this step, we were fortunate enough to make some NADH. So again, those are my high energy electron carriers. So they took some of the electrons that were liberated as those carbon-carbon-carbon-hydrogen bonds were broken down. And where did they go? The NADH are taking the high energy electrons to the electron transport chain. All right, so now what we're going to look at briefly is what's happening to the 2 2-carbon molecules as it's entering into the citric acid cycle. All right, so this is stage 3 of cellular respiration. We have a 2-carbon molecule, acetyl-CoA, and we've got 2 of those. And so they are going to enter into what is known as a metabolic cycle. So some molecules are going to enter and others will be leaving, but this cycle will continue on indefinitely. I'll show you what that looks like. So acetyl group is going to be removed from acetyl-CoA and is going to be attached onto a 4-carbon molecule known as oxaloacetate to form citrate or citric acid. And that's why this is called the citric acid cycle, also sometimes referred to as Krebs. Now, the key takeaways is that as we are taking our acetyl-CoA that enters into the cycle, as it goes through this cycle, what we're going to generate are 2 CO2s, 1 ATP, 3 NADHs, and 1 FADH2 for each acetyl-CoA. So that means we're going to make, for our two carbon molecules, we're going to make 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s. So this is a great time to get out your chart and go ahead and write this down. And the key is that oxaloacetate is going to continue to get regenerated and start the cycle all over again. So let's look at this. Here we've got oxaloacetate, which I said is a 4-carbon molecule, and here is our acetyl-CoA, and we've just attached on this sulfhydryl group and coenzyme A, but now we're going to literally take it off in the very next step. Now the reason for that is, as we break that down, that is going to provide a lot of energy in order for our 2-carbon molecule, which again originally came from glucose and then was a part of pyruvate, and now we've got our 2-carbon molecule that is going to be able to get linked, attached, bonded to oxaloacetate, and now we're going to make a 6-carbon molecule known as citrate. And now what we're going to do is just continue to break down and modify the citrate until we regenerate our 4-carbon molecule known as oxaloacetate. Okay, so the key is our 2 carbons from the acetyl-CoA are going to be lost in this process in the form of CO2. But as we make all these modifications and slight changes to citrate, we're also going to be generating 3 NADHs for each acetyl-CoA, an FADH2, and 1 molecule of ATP. All right, so you are not responsible for memorizing what is going on during the citric acid cycle. We want you to have a big idea of what we're generating, which is all the NADHs and the FADH2s and the ATPs. However, if you were so inclined, you could look at each of these processes and identify some of the key things that are going on. For instance, so here we have our acetyl-CoA, and it's being, and we immediately lose the coenzyme A and the sulfhydryl group. And here we've got our 2 carbons that are now attached to oxaloacetate, which was our 4-carbon molecule coming into this. 1, 2, 3, 4. So now we've got a 6-carbon molecule. And that's going to undergo some isomerization, and what you can see here is if you look at the specifics of what's going on in each of these steps. For instance, here we're going from a hydroxyl group to a double bonded oxygen. And so what happened to that hydrogen? Well, this is one of the steps where we are forming NADH. Again, if you can track your hydrogens, you can track where your electrons are going. So here we go from a 5-carbon molecule down to from a 6-carbon molecule down to a 5-carbon molecule, and we're going to be liberating a CO2. What happens to that CO2? You exhale the CO2. So again, we aren't going to expect you to memorize these structures. If you take biochemistry later on, you will get to do that then. But for all intents and purposes, what we want you to understand is that for our 2 acetyl-CoAs that came into this process, we are going to have a net production of 6 NADHs, 2 FADH2s, and 2 ATPs. And just to clarify, I've talked quite a bit about NADH, but FADH2 is also a high energy electron carrier, and it's just like NADH, it's going to be carrying those electrons to the electron transport chain and dropping the electrons off there. All right, so again, in summary, for each acetyl-CoA, we're making 3 NADHs, 1 FADH2, 1 ATP, and 2 CO2. So all of our carbons, by the end of the citric acid cycle that originally came from glucose, have now been all exhaled. Okay, so at this point, there's no more carbon carbon-carbon, carbon-hydrogen bonds to break down because it's all been lost in the form of CO2. However, we do have a lot of high energy electron carriers, namely the NADH and FADH2, that are now going to take those electrons to the ETC, which is where we will make a lot of ATP. The last thing I want to briefly cover is this idea of how would we possibly regulate all of these different metabolic pathways. How can we regulate this system, which is got so many steps? It's important to think about what is it that we're ultimately trying to make. How are we going to ensure that we aren't going to keep driving all of these enzymatic processes forward if we don't need to? So I want you to pause the video here for a second and answer on the Ed puzzle. How do you think we can ensure that we are not going to constantly be going through these processes if the energy is not needed? What do you think might be an appropriate inhibitor of these pathways? If you think about it big picture, what's the goal of breaking down glucose? Well, it's really to make ATP. It's really to generate this stored form of energy that can be used to drive other endergonic reactions. But what if you don't need the energy? What if you have a lot of ATP? Then we're going to want to shut these pathways down. And how do you think we could shut down the pathways? How could we inhibit different enzymatic steps? Well, the key is going to be ATP. Interestingly, if we have enough ATP in the cell, ATP can directly act as an inhibitor of some of the key enzymatic steps, going all the way back to glycolysis itself. What we can see here is that one of the earliest steps in the breakdown of glucose is this enzyme known as phosphofructokinase. And this is directly inhibited by ATP. It can act as an allosteric inhibitor, meaning it binds not at the site where the substrate binds, but at an alternative location, but it will disrupt the substrate binding site such that fructose 6-phosphate can no longer bind. And if the substrate cannot bind to the enzyme, then we will not be synthesizing this product. We see that in Krebs cycle, it likewise is inhibited by high levels of ATP. Again, if we have enough ATP, we don't need to go through the process of trying to break down glucose in order to make ATP. So we see early steps such as citrate synthase is inhibited by high levels of ATP, as well as high levels of NADH. All right, so we just did a lot together. We went through the breakdown of glucose, our 6-carbon molecule, into 2 3-carbon molecules known as pyruvate. In that process, we were able to generate 2 ATP as well as 2 NADHs, which are going to shuttle those electrons to the ETC. Our pyruvate, our 3-carbon molecules, of which we had 2, were then transported into the inner mitochondrial matrix, where they were synthesized, broken down, and made into acetyl-CoA. We lost 2 of those carbons off of our 3-carbon molecule. The 2-carbon molecule, acetyl-CoA, then entered into the citric acid cycle, where we further broke down those 2-carbon molecules until ultimately all of our carbons at this point were lost and exhaled into the environment. In the process of the formation of acetyl-CoA, we did not make any ATP, but we did make 2 NADHs in that step. During the citric acid cycle, likewise, we made 6 NADHs and 2 FADH2s. So now entering into the electron transport chain, we have 6 NADHs from step 3, 2 from step 2, and 2 from step 1. So we have 10 NADHs that are going to be dropped off at the ETC, and 2 FADH2s. At this point, all of our carbons have been oxidized and exhaled, but now we're going to go ahead and make the bulk of our ATP at the electron transport chain. All right, so in summary, you should be able at this point to answer these 6 learning outcomes that we proposed for you in the beginning of this section in the overview on Canvas. So first of all, you should be able to outline the 4 metabolic stages involved in the breakdown or the oxidation of glucose into CO2. You should be able to describe the role of NADH and FADH2 during cellular respiration. You should be able to calculate the substrates and products, meaning NADH, FADH2, and ATP, during these 4 stages in cell respiration, and where are they all taking place. You should be able to outline the 3 phases of glycolysis and what the key steps that are happening there. How is it that pyruvate gets oxidized? Or you could also say, how is it that acetyl-CoA is formed? And then lastly, are you able to explain this concept of a metabolic cycle? How does that relate to the citric acid cycle, sometimes called the TCA, or also known as Krebs? What are the net products produced in that cycle, and how is it ultimately regulated? I hope this was helpful. If you have any questions, please let myself or Dr. Durrell know, and we are happy to help.